Hi, this is Mr. Hagee, and I'm going to discuss with you the setup and procedures for our photosynthesis lab. In part one of the photosynthesis lab, we're going to be measuring the rate of photosynthesis in plant leaves using white light. And in part two of the lab, we'll be measuring the rate of photosynthesis in plant leaves using different colors of light. So let's take a look at the apparatus for measuring our photosynthesis. So in our setup, we'll have a external light lamp here with a clear 200 watt bulb. Next we have here is our heat sink, and the heat sink is a 4-liter aquarium full of water, just cold tap water. And the heat sink is here to absorb any heat that may be coming off that is going to be coming off the light bulb, because we do not want heat to affect the results of our manometer movement. We have test tube rack, as you can see, with a test tube, and this is a 25 by 150 millimeter glass test tube, Pyrex tube. The manometer that we're going to be using to measure the rate of photosynthesis is one that I've designed here, and it's basically a plastic tubing with a U-shaped manometer in it. And then we have transparency graph paper that is measuring in 5 millimeter increments. And then we have a straight tube, plastic tube with a rubber tube attached to it that is currently open to atmosphere. And then we'll talk about how we're going to use this manometer here shortly. The plant leaves that we're going to be measuring the rate of photosynthesis in today will be collard greens. Okay. And we're going to have to measure out 2 grams of the collard greens here in a second. And our carbon dioxide source, we're going to be using 0.5% sodium bicarbonate. Okay, so let's talk about the first thing we need to do is add the manometer fluid to the manometer. And the manometer fluid is really just glycerol and water with a little green food coloring. So the first thing you'll do is add the manometer fluid, and it's a little easier to use a plastic pipetter than the glass pipetter that comes with the bottle. And you're only going to need a little less than or around a half a milliliter or so. And so put it down the tube, and you're just going to add it to the tube, and we're going to need a little bit more, as you can see here. So now it's going to be a little trickier, because as we add more, it's going to push that up. So what we need to do is push it down, back and forth here to get the fluid to join together and settle in the manometer. Okay, so we need to make sure we have enough that comes around the bend here and is up on our graph scale. So we need to add a little bit more. Again, we need to make sure that there are no air bubbles, that all the fluid is uniformly within the tube here. No air bubbles. Alright, so that looks like that's going to work there. Next thing we're going to do then is measure out our 2 grams of collard greens. Scale. Okay, let the scale go to 0, put on the weigh boat, and make sure you zero out the weigh boat. And then it's just as easy, you just tear off little pieces of the collard green until we get 2 grams. And you can try to keep the size of the pieces relatively small here. 1 gram. 2 grams here. Exactly. So now we need to add our color greens to the test tube. Now we're ready to add our sodium bicarbonate. You need to add enough sodium bicarbonate so that we leave about a centimeter or 2 centimeter airspace at the top. We do not want the stopper to come in contact with the fluid. And again, if the inside of the tube gets a little wet, you might take a paper towel and dry that out, so make sure you get a good seal here. All right. Okay, so that's the setup is really pretty quick, and so now we're ready to do our first run. First thing, you need to make sure that the plant leaves are 20 centimeters from the light source. So use your half-meter stick here and make sure the center of the tube is at 20 centimeters away from the light source. And then we're going to turn on the light, and we have a 5-minute equilibration period with the straight tube here unclamped. Okay, so just turn on the light, and then we also will shut off all the room lights. Okay. And so you'll start your timer. You can have your cell phones out and start your timer, and we want to let it set for 5 minutes. Unclamped. Then after the 5-minute period, we're going to start our first run. And to start our first run, we'll take the Hoffman clamp here and make sure that the rubber tubing is all the way to the back edge of the clamp here, and then we'll clamp it off. Okay. Now before you clamp it off, you need to take your transparency pin and mark the level that the manometer fluid is at the start. And so we'll just take and make sure you get down here to eye level, so you get an accurate marking of the level of the fluid and the transparency fluid on the transparency. And then when the 5-minute equilibration is up, you'll close off the straight tube here. And once you do that, it will now have created a sealed chamber. And so as photosynthesis occurs, it's going to produce a gas, right? Oxygen gas, and which will cause the manometer fluid to rise now that we've created a sealed chamber here. You'll let this run for 10 minutes. And at the end of 10 minutes, you will then unclamp, at the end of 10 minutes, you'll mark the level that the fluid has risen on the graph paper. And then you'll unclamp the tube and let it back down to atmosphere. The fluid will move back down to the starting level. And then you'll wait 3 minutes. And after 3 minutes, you'll start another 10-minute run, and you'll clamp off the straight tube and start your timer and do a 10-minute run. After the second 10-minute run, you'll unclamp it, let it back to atmosphere, wait 3 minutes, and then do a third 10-minute run. And then that is part 1 of this photosynthesis lab.